previously, we have learned about passive transport, which is the diffusion of a substance across a biological membrane down its concentration gradient. But what happens when a substance needs to be transported from an area of low to high concentration? Can a cell even do that? Such situations require what we call the active transport. By the end of this video, you can describe how active transport is accomplished, and you can as well define the various forms of bulk transport that can move materials into or out of a cell. You might ask, how come energy is not required for passive transport, like mentioned in the previous video, but is a must for active transport to happen? Take this illustration for an example. Rolling a rock down a steep slope do not ask for you to expend energy, just like how effortless you would go down a slide. On the other hand, if you were to put back the rock from the ground to the top of the hill, you will need a lot of energy and assistance to bring the heavy rock up. I hope that this analogy provides a clearer view in explaining the difference between energy requirements of passive and active transport. Now, don't get confused with facilitated diffusion, which makes use of membrane proteins. Despite the help of transport proteins, facilitated diffusion is considered a type of passive transport because the solute is moving down its concentration gradient, a process that requires no energy. Facilitated diffusion speeds transport of a solute by providing efficient passage through the membrane. But it does not alter the direction of transport, unlike that of an active transport. So how does active transport happen? To pump a solute across a membrane against its gradient requires work, and the cell must expend energy in the form of ATP. Recall that ATP means adenosine triphosphate. This means that it has three phosphates attached to it. When a phosphate group is removed from ATP, it becomes ADP, which means now as adenosine diphosphate, having only two phosphates. The phosphate group that binds to the protein causes it to change shape and deliver ions across the membrane. Active transport enables a cell to maintain internal concentrations of small solutes that differ from the concentrations in its environment. One example of an active transport system in the membranes of most animal cells is what we call the sodium-potassium pump. This uses ATP as an energy source to expel 3 sodium ions for every 2 potassium it admits. An animal cell, for example, should maintain a higher concentration of potassium ions and a much lower concentration of sodium ions. Let's take a look at diagrams 1 to 3. For you not to be confused, let's focus on sodium. Sodium ion is high outside the cell and low inside. As mentioned, the cell needs to maintain a low concentration of sodium inside. That is why the cell will expel sodium ions outside even if there is an already low level of sodium inside. To do this, energy is needed. So when the cytoplasmic sodium bind to the carrier protein, a phosphate group from the ATP binds to the carrier protein and ATP becomes ADP. This phosphate, symbolized by letter P, will serve as a token to power the protein to change in shape and expel the sodium ions outside the cell. Now let's examine diagrams 4 to 6. In the case of potassium ions, its concentration is low outside the cell and high inside. Again, cells need to maintain high concentration of potassium inside the cell, so it will want to receive even more potassium ions from a low concentration outside going in. Continuing where we left off, Potassium ions from outside the cell bind to the same carrier protein and triggers the release of the phosphate group attached earlier. When the phosphate was removed, this caused the protein to change back to its original shape, and the potassium ions it carries are taken inside. Now, both animals and plants experience active transport at the cellular level. Other examples of active transport include ions moving from soil to plant roots, minerals traveling through a stem to various parts of the plant, sugars from photosynthesis moving from leaves to fruit, amino acids moving along the human intestinal tract, glucose moving in or out of a cell, enzyme secretion and release of antibodies. Moving on, let's have the last example of membrane transport, which is the bulk transport. Water and small solutes enter and leave the cell by diffusing through the lipid bilayer of the plasma membrane, or by being pumped or moved across the membrane by transport proteins. However, large molecules such as proteins and polysaccharides and other larger particles generally cross the membrane in bulk, packaged in what we call the vesicles. This type of transport is called the bulk transport. And like the active transport, these processes require energy. There are two types of bulk transport as you can see in this diagram. We have the exocytosis, which exports large quantities of material from the cell going out, and endocytosis, which is the export of large quantities of material from outside going in. An example of bulk transport is seen in the transport vesicles that buds from and to the Golgi apparatus. There are two sides of the Golgi bodies, the receiving side and the shipping side. The receiving side of the Golgi apparatus is an example of endocytosis because it takes in the materials given by the endoplasmic reticulum. On the other hand, exocytosis can be seen on the shipping side of the Golgi. Some of the membrane of the Golgi bodies butt off and form transport vesicles. These act as packages to be delivered outside the cell. When the membrane of transport vesicles from the Golgi and the plasma membrane come into contact, the two membranes fuse. Let's take a closer look at endocytosis. In fact, the events of endocytosis look like the reverse of exocytosis. And there are three types of endocytosis, and the one you're seeing is phagocytosis, or also known as cellular eating. In phagocytosis, a cell engulfs particle by extending pseudopodia, or pseudopodium if it's just one, around it and package it within a membranous sac called a food vacuole. Pseudopodium is a temporary protrusion of the cytoplasm of a cell for movement or for feeding. This can be observed in the organism amoeba, such as the one shown here, or even cells residing in our body, such as the white blood cell. The particle will then be digested after the food vacuole fuses with a lysosome containing hydrolytic enzymes. Next, in pinocytosis, a cell continually gulps droplets of extracellular fluid into tiny vesicles formed by infoldings of the plasma membrane. To explain, extracellular fluids are fluids not contained inside the cells or simply the environment outside the cells. This environment of cells carries micronutrients that can be used by the cell. Going back, the cell obtains molecules dissolved in the droplets. Now, what are these materials that coat the vesicles? This one. In many cases, the parts of the plasma membrane that form vesicles are lined on their cytoplasmic side by a layer of coat proteins called pits, and the resulting vesicles are said to be coated. An example of pinocytosis is observed in the microvilli of the small intestine to absorb nutrients from the gastrointestinal tract. The cells that line our intestines look like protrusions to increase the surface area for the absorption of food. First, tiny droplets of fluid are trapped by the folds of the plasma membrane. Next, these folds in the plasma membrane pinch off into the cytosol as vesicles. Last, contents of these vesicles are then slowly transferred into the cytosol. The last type of endocytosis is the receptor-mediated endocytosis. Embedded in the plasma membrane are proteins with receptor sites exposed to the extracellular fluid. Specific solutes bind to these receptors. Since receptor-mediated endocytosis is a special type of pinocytosis, you expect to find a cluster of coated pits. Each coated pit forms a vesicle containing the receptors with molecules from the extracellular fluid bounded to it. The diagram shows only bound molecules such as these purple triangles inside the vesicle, but other molecules from the extracellular fluid are also present, like the green circles here. After the ingested material is liberated from the vesicle, the emptied receptors are recycled to the plasma membrane by the same vesicle. As an example, we have here an LDL, also known as the bad cholesterol, due to the risk for heart disease and stroke. So this cholesterol attaches to the receptors in coated pits of the plasma membrane. Next, a vesicle is formed inside the cell containing the LDL. The proteins involved in the coated pits go back to the plasma membrane to assist in another possible round of endocytosis. The vesicle is then met by endosomes and lysosomes to digest this cholesterol for the cell to use. The receptors will then go back to the plasma membrane to do its function again. Let us now summarize the key concepts of active and bulk transport. In active transport, a carrier protein uses energy in the form of ATP to move a substance against its concentration gradient. For example, the sodium-potassium pump uses active transport to exchange sodium ions for potassium ions across an animal cell membrane. In bulk transport, on the other hand, it requires vesicle formation and metabolic energy. It occurs independently of concentration gradients, depending whether it's high to low or low to high. Exocytosis transports macromolecules out of a cell via vesicle formation and often results in secretion. Endocytosis, on the other hand, is the transport of molecules into a cell via vesicle formation as well. And receptor-mediated endocytosis makes use of receptor proteins in the plasma membrane. Now, all types of membrane transport are essential for cellular life. As cells proceed through their life cycle, a vast amount of exchange is necessary to maintain function. This is made possible by the different transport mechanisms that incorporate biological molecules and discharge waste products necessary for normal functioning of the cell.